Thank you, organizers, for the opportunity to give a talk today in this seminar series. In the past, starting from 1993, I spent 5 years in Japan and 6 years in Edmonton in basic research, focusing on islet isolation and transplantation. I know the majority of the audience are basic researchers, but I'm not going to talk about basic research today because I'm not currently doing any basic research. Okay? But I hope you can learn something from my talk. So, over the next 20 minutes, I'm going to talk about the past and current activities of the Clinical Islet Laboratory at the University of Alberta Hospital. First of all, I would like to briefly explain islet isolation and transplantation for those who may not be familiar. The pancreas is retrieved from a deceased donor, transported in a cooler to our clean room facility. Upon arrival, the pancreas is trimmed and distended with cold enzyme solution through the pancreatic duct. Then the pancreas is enzymatically and mechanically digested. Next step is purification. This is done using continuous density gradient with a centrifuge machine. Purified islets are collected and counted. At this stage, when we obtain enough islet for transplant, we initiate the transplant process. Meanwhile, islets are preserved in an incubator. On the day of transplant, we do release tests, including a final islet count. Transplant preparation is brought to the hospital. This is Peter Sr., who is an endocrinologist. Transplant procedure is easy enough, so he can do this. We have done 711 islet transplant into 304 patients with type 1 diabetes since 1999. Regarding islet isolation procedure, it takes about 5 hours from starting islet isolation to this stage. Preservation period or culture time is less than 72 hours, average about 30 hours. Importantly, we have to be always ready to prepare islet for transplant for 24 hours a day, 7 days a week, and 365 days a year. Now, I'm going to show you a video. Ambulance brings the pancreas from the airport. I have to scrub in because I touched the pancreas. The pancreas with spleen and duodenum. This is a cannulation procedure. The pancreas is distending with enzyme solution. The pancreas is now into the Ricordi chamber, starting digestion. We take a sample during the digestion and assess digestion progress, collecting tissue, pre-purification counting. Now, purification step. Loading pancreatic digest into a centrifuge machine. This is a continuous density gradient. Counting islet, always two independent counters, and viability assay. We preserve islet in an incubator. On the day of transplant, islets are transferred into a transplant bag. Now, ready to go to the hospital, 10 minutes walk distance. A needle is in the main portal vein under ultrasound guidance. A guide wire is in the main portal vein. A catheter is located in a good position. Now, ready for infusion. Starting infusion. This shows the activity of islet isolation and transplantation over the 38 years at the University of Alberta. White bar indicates the number of islet isolations per year, and the black bar is the number of transplant procedures per year. First of all, I want to show you what was done before Edmonton Protocol. The first human islet isolation was performed in 1984. During the pre-Edmonton Protocol era, 7 islet transplants were performed. This gentleman became the first islet transplant recipient in Canada in 1989. You may recognize Ray Lajot and Gus Warnock here. The number on this sign held up by Ray Lajot is 100. It took 100 human islet isolations to perform the first transplant. Great efforts were made to perform the first transplant. Here is a summary of 7 cases. During those days, islet transplant was considered an experimental procedure. Therefore, islet transplant was relying on other organs, mostly on kidney transplant. Also, it was difficult to isolate enough islet mass from a single donor pancreas. In order to obtain critical islet mass, cryopreserved islet from multiple donors, as many as 10 donors were combined. As a result, case number 3 and case number 5 achieved insulin independence. And the long-term outcomes of these 2 patients were reported in cell transplantation. Now, during this time of period, clinical islet transplant was not performed due to funding and other logistical issues. However, 122 human islet isolations were performed during the 3 years. My mentor, Ray Lajot, often said, "Complete your study, don't stop it." He didn't stop islet isolations during this difficult time. I moved to Edmonton from Japan in June 1998, and as a privilege to work for the development of Edmonton Protocol with many pioneers at the University of Alberta, including Ray Lajot, Gus Warnock, Jonathan Leakey, Greg Corbett, Eddie Ryan, and James Shapiro. The first transplant under Edmonton Protocol was done on March 11th, 1999. Just 9 months after I joined the clinical islet isolation team. It was home run.
A major breakthrough in the war on diabetes.
Canadian scientists have succeeded in giving test patients cell transplants,
a breakthrough treatment that could mean a better quality of life for millions.
Insulin independence in 7 consecutive patients with type 1 diabetes. I was very fortunate to be a member of islet isolation team from the beginning of Edmonton Protocol. Back to this slide again. What I would like to emphasize by showing this slide is that tremendous and continuous effort were made before Edmonton Protocol. Edmonton Protocol was not developed in one day. Well, insulin independence using a single donor is the most desirable goal of islet transplant, but not a primary aim of islet transplant. However, in my opinion, we should stick with insulin independence. 20 years ago, achievement of insulin independence after a single islet transplant was not common. The rate was less than 20%. In the recent 3 years, almost half of the recipients became insulin independent after a single infusion. This improvement is largely due to a higher islet mass transplanted. Indeed, 20 years ago, I was so happy when I got 300,000 islet equivalent. Nowadays, 300,000 does not make me excited at all. This paper was published in 2014. As you can see from this Kaplan-Meier curve, insulin independence does not last long after a single transplant, unfortunately. Good news is that this patient is still enjoying insulin free after a single transplant. It is now 13 years. Again, islet mass is critically important in graft outcomes, and functional assay does not give us useful information. One thing I want to mention here is that we set this bar at 5,000 IQ per kilogram body weight as a minimum islet mass for transplant 23 years ago. I would like to draw your attention to the fact that there have been no changes in this bar for 2 decades in spite of improvement in islet isolation outcomes. I'm confident that we can set this bar higher, for example, 6,000 IQ per kilogram body weight. This shows the disposition of islet preparations since 2007 when I took over my previous supervisor's position. There are some fluctuations in transplantation conversion rate, and the overall conversion rate is 46%. This clearly indicates that we still have a room for improvement in islet isolation outcome. We also distribute human islet for research when we don't get enough islet for transplant and research consent is given. About 36% of preparations were distributed for research. I don't have enough time to talk about Alberta islet distribution program today, but if I have a chance, I'd like to talk about the program. Intraportal transplant is often criticized because of several reasons, including instant blood mediated inflammatory reaction. Extrahepatic site has been a hot topic in islet transplant community. We have conducted 3 clinical trials investigating extrahepatic site. However, results are not satisfactory. First one was subcutaneous islet transplant using an artificial pouch. Insulin positive cells were detected in histological examination, but no graft function, no C-peptide secretion was detected at all in all 3 patients. Then we tried gastric submucosal transplant in 2 patients. We observed some degree of graft function in both patients, but no insulin independence was achieved. Lastly, we performed omental pouch transplant in 4 patients. Again, we observed reasonable graft function, but no insulin independence was achieved. Extrahepatic site is attractive, but I can conclude that intraportal islet infusion remains the gold standard. In addition to the 3 extrahepatic trials, we have done many other clinical trials. This is a busy slide, but just a list of clinical trials in your program. These 2 trials are currently ongoing. In some clinical trials, intervention was done within clinical islet laboratory. For example, in these 4 trials, prior to islet isolation procedure, we perfuse the pancreas with solution containing anti-aging glycoprotein, antioxidant agent, alpha-1 antitrypsin, or caspase inhibitor. Here is an example of vascular perfusion. This case is anti-apoptotic agent caspase inhibitor. This is a superior mesenteric artery and splenic artery. The flow rate is automatically adjusted to maintain vascular pressure using a dual pump system. Now, I switch a gear to the pancreas donor. In Canada, the number of donors is increasing, nearly triple increase during a 10-year period, as shown here. DCD means donation after cardiac death. Both branded donor and DCD are increasing. At the same time, we are declining more donor offers. Why is that? Well, bottom line is that we don't have enough recipients in our waiting list. Many donors are declined because of no ABO compatible recipients or no virtual cross-match negative recipient. I don't think I have to explain ABO compatibility here, but I will explain virtual cross-match in a minute. Previously, we had many recipients across Canada, but in 2007, Alberta government decided not to accept patients who live outside of Alberta because of financial issue. As a result, our recipients are limited in Albertans with a few exceptions. This is ironic because we accept donors across Canada, but no recipient from outside of Alberta. Here is a snapshot of our recipient list. There are 1 blood type A and 6 blood type O potential recipients with no Bs and no ABs. One day, we had an offer of this blood type O donor. All recipients except for one are virtual cross-match positive. For example, this recipient has antibody against HLA-B7, probably due to pregnancy and or previous transplant. This donor has B7, so for this case, virtual cross-match is positive, and this recipient is not eligible. The same is true for the other highly sensitized recipient. Only this non-sensitized recipient was eligible because virtual cross-match was negative. We accepted the donor, isolated Islet, and then transplanted. What happens next is that we will have to continue to decline many donors until a non-sensitized recipient is newly listed. This recipient shortage is a chronic problem and is an ongoing issue. This is a Kaplan-Meier curve showing how quickly patients undergo transplant once listed. If patients are not sensitized, waiting time is 1 to 2 weeks. But if patients are highly sensitized, they have to wait years and years. Okay, this is my last topic. As you may know, total pancreatectomy is performed for chronic pancreatitis when other treatment methods are not successful. To reduce a risk of pancreatectomy-induced diabetes, autologous islet transplant is performed worldwide. Resected pancreas is brought to islet isolation facility and islet isolated and then infused back into the portal vein of the same patient. We have done total pancreatectomy and autologous islet transplant in 73 cases since 2002. We performed this procedure in 9 children with gene mutation, including a 2-year-old girl. This is the youngest case reported in the world. We all know if you fail to cannulate the pancreatic duct, you don't get any islet at all. For this case, it was especially difficult to cannulate a tiny duct of extremely inflamed pancreas. Indeed, this case was the most difficult and stressful islet isolation for me among more than 2,000 cases I performed. In summary, tremendous efforts have been made before Edmonton Protocol. Intraportal infusion is still the gold standard. Recipient shortage is chronic and ongoing problem. We continue to be involved with clinical trials. I would like to thank to my fantastic team members, especially to Doug O'Gorman. He has been working at the clinical Islet laboratory for more than 20 years and has been a great team leader. Every year on Canada Day, I tried to take a family picture. I had been on call every day until 2017, so I had to cancel family events many times, but my wife Noriko never complained. I would like to give special thanks to her. Thank you for your attention, and I'm happy to take any questions.